Okay, now we're going to discuss cytogenetics, which is the study of chromosomes. The chromosomes are defined as DNA molecules and the associated proteins. Most of this comes from chapter 8. Prokaryotic chromosome structure, though, comes from chapter 2. Molecular structure of eukaryotic chromosomes from chapter 11. And then chromosome breaks, unequal crossing over aneuploidy and polyploidy. All that comes from chapter 8, as do the problems you will be working. So prokaryotes have a single circular chromosome, and it's wadded up in the central part of the cell, as we can see in the top image there. And that's what the DNA is labeled. It's the chromosome. Here's the genome and the chromosome of the first organism to ever have its entire genome sequenced, Haemophilus influenzae. That was done back in 1995. And at 1.3 million base pairs, it's pretty small relative to eukaryotic chromosomes and genomes. Prokaryotes divide by binary fission, and of course the chromosome has to be replicated. Before that, we see two origins of replication, indicating that in that little mess there in the second from the top image is really two chromosomes, and they separate as the cell divides. Onto eukaryotes. Now, chromatin is DNA in the interphase of the cell cycle. It's not condensed into chromosomes. It has a bead on a string-like appearance. Beads are nucleosomes, which are DNA wound around histone proteins. So here's showing the cell cycle. That M phase, that's the only time chromosomes are in existence. During interphase, the DNA is in the form of chromatin, not chromosomes. So we see a chromosome, unduplicated one at the top, a duplicated one below that. So when cells enter into cell division, the chromosomes are duplicated because they've just gone through DNA replication. And so the two sister chromatids, remember, are copies of one another. And this shows the basic anatomy of a eukaryotic chromosome, with the main constriction being the centromere, spindle fibers attached to the kinetochore, and then telomeres cap the ends of the chromatids. So these nucleosomes then are DNA, which is here shown in green, wrapped around an octamer of histone proteins, which is shown in pink there, with an additional histone protein clamped on the outside of each nucleosome. The DNA in between the nucleosomes is referred to as linker DNA. Here's a close-up of a nucleosome with the DNA in white, and then the different histones making up that octamer shown. It's made of two histone 2As, two histone 2Bs, and a histone 3 in blue there, and a histone 4 in green. You just have to know what a nucleosome is and what histones are. You don't have to know the names of all the histones, though. Here's a photomicrograph of chromatin showing you the nucleosomes as little beads there. Now, moving into the structure of eukaryotic chromosomes. The nucleosomes coil to make a fiber 30 nanometers in diameter. And then this is further condensed into fibers that are 250 nanometers in diameter that loop in and out of the scaffolding protein that makes up the backbone of the chromatid. Mainly that's topoisomerase 2. And we think the genes are located on the loops of the DNA that stick out of the scaffolding proteins. So this shows you right from your textbook the ways that DNA is packaged into a chromosome. And then this happens during prophase of mitosis and meiosis as the cell is getting ready to divide. If we add nuclease, which is enzymes that degrade DNA, we see a little bit of nuclease will degrade some of the linker DNA, splitting up the individual nucleosomes, and even more nuclease added will get rid of the rest of that linker DNA, and we see that somewhere about 146 base pairs are wrapped around each histone octamer in chromatin. Here's a human metaphase chromosome. Usually you see chromosomes from metaphase because they're at their most condensed, but we can see the 250 nanometer fibers looping in and out of the chromatid, and we think that's where the active genes are. So karyotype is a picture of the chromosomes. Here they are stained in a certain way so we can match up the homologs. And this, of course, is a human male, as we can see with the X and the Y chromosome there. The autosomes tend to be numbered with 1, the largest, and the smallest gets the highest number, although in us humans, actually 21 is a little smaller than 22, but we didn't know that when they were originally numbered. And we can stain chromosomes in different ways to match them up and identify them. G-banding is a very important way that shows AT-rich regions in the dark bands. And G comes from Giemsa, the word Giemsa, which is a type of stain that you use to stain the chromosomes after they've been treated with various chemicals. Q-banding shows AT versus GC-rich regions of the chromosomes when viewed with UV light. C-banding shows heterochromatin, so the dark regions are the heterochromatin that I've defined before, the inert DNA. Mostly that's centered around the centromeres, but not in all creatures. R-banding is a way to show GC-rich regions of chromosomes. You can see the red and yellow banding when viewed under UV light. And so that's all I've got on the different ways that chromosomes can be stained. So anyway, now we're going to discuss chromosomal breaks and how that affects heredity. Now, mutations are any change in the DNA molecule. Gene mutations are small ones that involve individual base pairs or small amounts of base pairs, small amounts of base nucleotides. But we're going to talk about in this unit chromosomal mutations, that is breaks of the chromosomes or changes in the numbers of chromosomes. These breaks can be spontaneous. Any environmental agent that causes a mutation is called a mutagen. And then a class of mutagens that break chromosomes are clastogens. These include things like X-rays, physical and chemical stress to the cells and the chromosomes. This shows the basic types of chromosomal rearrangements. So duplication is the duplication of part of the chromosome. Here we start at the top with genes A, B, C, D, E, F, G, and we can see here in the rearranged chromosome, genes E and F have been duplicated. The deletion is missing genes, where E and F have been deleted. And then inversions are where the chromosome breaks in 2 places and the region between the breaks flips over 180 degrees. And then translocations are usually exchanges of DNA between non-homologous chromosomes, as shown at the bottom there. Telomeres then. These cap the ends of unbroken chromosomes. They're heterochromatic, and they have their, of course, DNA sequences with associated proteins. But when chromosomes break, there's no telomere there, and enzymatic processes in the cell want to stick stuff there, so we call those sticky ends. And that's why you get things like translocations occurring. And sometimes the wrong segment is there, and that's when a translocation happens. Restitution is the process by where the right piece of DNA is stuck back on where it broke off from. So we recognize single and double chromosome breaks. 2 types of single chromosome breaks are referred to as chromatid and chromosomal breaks. And then the double breaks we'll discuss are inversions and translocations. So we'll discuss the single breaks first and then the double breaks. So a chromatid break occurs after DNA replication, so the break is not replicated, so therefore the break is only on a single sister chromatid. Chromosomal break, though, break happens earlier before DNA replication, and now both sister chromatids have the break. Here's a chromatid break, and that yields a centric fragment. That is a fragment with a centromere when this thing splits, and then an acentric fragment. That's a fragment of the chromosome with no centromere. So then the best outcome would be restitution. But anyway, if that doesn't happen, we have 2 chromosomal fragments, the acentric and centric fragments like I just showed you. The acentric fragment will be lost because it has no centromere for the spindle fibers to attach to. And the centric fragment then will have a mutation called a deletion. And when we talk about that, we'll talk about that next. So here You can see two homologous chromosomes above. One has had a deletion, so it's a lot shorter than its pair. So the genes E and F have been deleted. So during meiosis then, so that the gene G's can line up correctly, the ones at the end, a bulge is produced in the normal chromosome because genes E and F don't have anybody to pair with, since there are no genes E and F on the other homolog. Okay? And so we've banded chromosomes and we can tell about their structure. So here we see bands 3C2 through 3C11. Those have been deleted on the lower chromosome, and that's why they're bulging out on the upper chromosome. And we can see a real deletion bulge on these Drosophila polytene chromosomes. In flies, and then in their salivary glands, their chromosomes will duplicate a lot and not separate. So you get these big, thick chromosomes, and those are what polytene chromosomes are, and that makes them pretty easy to study under the microscope. So what can be the effects of deletions? One is pseudodominance. We may only have one copy of certain genes, therefore greater likelihood of recessive alleles being expressed, which are often cause of diseases. Depending on which genes are lost, the deletion may be a lethal mutation if the important genes are lost. This is the wingtip of a normal fruit fly wing. And here's the wingtip, the notched wingtip of a fruit fly with a deletion. So the deletions do cause phenotypic effects. That's what this is showing. These seem to be fairly minor phenotypic effects, however. I assume the flies with this can still fly. On to chromosomal breaks. Here the break is replicated because it happens prior to the S phase of the cell cycle. And of course the acentric fragments can be lost at cell division. The sticky ends of the sister chromatids may fuse, forming a chromosome with two centromeres called a dicentric bridge. And this dicentric bridge may or may not be broken during cell division. So anyway, here's our break that occurs prior to DNA replication, and the break is replicated. And so now we've got a break in both sister chromatids at the same spot. And these broken ends may fuse together because remember, they are sticky ends, forming this thing called a dicentric bridge, a chromosome with two centromeres, and it may enter this breakage fusion bridge cycle where it breaks and then reforms and then breaks again in between cell divisions. And this usually results in the death of that cell line in a few generations. So here's our dicentric bridge. Sometimes the daughter cells may play tug of war with this thing. And if it doesn't break, the whole thing is left out of the daughter cells. And if it does break, it just depends on where the break occurs as to what deletions or duplications may be present in the broken chromosomes. And then it may reform again before the next cell division. Now on to double breaks. Here, chromatid breaks at 2 points. This can result in deletions, inversions, and translocations. Inversions result when the piece between the breaks flips over and reattaches, and then translocations remembers when a piece of DNA sticks onto a non-homologous chromosome. So we'll discuss inversions first. Two types are recognized: pericentric, where the inverted region includes a centromere; pericentric, where the inverted region does not include the centromere. And this can result, of course, in the reversal of gene order, position effects, where if you change the position of a gene, you change its expression. It can also disrupt what are called linkage groups or co-adapted gene complexes, but it may create new ones as well. And it can result in heterozygotes, that is, individuals having one normal and one inverted chromosome being roughly half sterile, half of their gametes are inviable. So here is a breakpoint between genes B and C and E and F, and then we have that region flip over. So now our gene order is A, B, E, D, C, F, G, H. And this is called a paracentric inversion because the centromere, the white ball there, was not involved in the inversion itself. So this shows peri and paracentric inversions, the difference between them. So you'll need to know that. What are position effects? Disruption of linkage or linkage groups can alter the expression of a gene. So if we change the arrangement of genes, or the linkage arrangement, we can change their gene expression. And an example comes from female Drosophila fruit flies that are X plus XW for the white eye allele that we talked about earlier. These flies, based on their genotype, should have a white eye, but because of an inversion in the X plus chromosome, they have a variegated eye color. The eye is both red and white. So the eye color gene has moved from towards the end of the chromosome, lies next to the centric heterochromatin, and that alters its expression, as we can see here. So this fly should have a wild type or totally red eye, but as you can see, it's mostly white with just a few red facets, or the facets are called ommatidia, by the way. And here we see the top chromosome in B here normal. After it's had its inversion, the W plus gene for the color of the eye has moved next to the green stuff, which is the heterochromatin near the centromere, and that alters its expression. So that's called a position effect. You change the position of the gene and you alter its expression. What about the meiotic consequences of having an inversion? Well, inversion heterozygotes show semi-sterility because they have problems with crossing over. In a paracentric inversion heterozygote, a single crossover produces two non-recombinant chromosomes, which will produce viable gametes, but one dicentric bridge and one acentric fragment, both of which will produce inviable gametes. Pericentric inversion produces two non-recombinant chromosomes, which will produce normal gametes, and two imbalanced chromosomes. That means chromosomes with deletions and duplications, and they will produce inviable gametes. So here we can see at the top two or a pair of homologous chromosomes. The lower one has had an inversion. The inverted segment there is shown in the little black box. Now, in order for these guys to undergo synapsis, the chromosome with the inversion has to loop around on itself. And of course, that's called an inversion loop. And now we can see that the genes are all lined up properly. But the problem comes if there's a crossover within that inversion loop. Here's a real inversion loop, some polytene chromosomes. This is an inversion loop, again, with a crossover between gene C, the yellow gene, and gene D, the green gene. And here's an inversion loop in a pericentric inversion, again with a crossover between genes C and D. Now we see that the centromere is in the inversion loop in a pericentric inversion. Moving on with our paracentric inversion, here are the results in the gametes down here. The top chromosome is normal, the next two are inviable, and then the bottom one has the inversion, but it is still producing viable gametes. So half the gametes are inviable, hence the term semi-sterility. So the evolutionary consequences here, if half of your gametes are inviable, that probably will reduce your fitness, your reproductive success. Inversions can create or destroy these linkage groups or co-adapted gene complexes. So they could be beneficial mutations at times, but usually they are thought to be deleterious mutations. This shows our paracentric inversion from start to finish. So in a problem, I would give you the inversion loop and expect you to tell me this stuff at the bottom. And those two things in the middle, we can kind of ignore those. Those are just intermediate steps. For a pericentric inversion, we get similar but not identical results. Again, the inverted part is in the black box there, black rectangle. So our gametes aren't as drastically messed up as in a paracentric inversion, but we can see the two in the middle there are inviable because of deletions and duplications of genes, and the ones on the top and the bottom have all the genes, and the one on the top is a totally normal one. The one on the bottom has the inversion, but that's okay. It's still going to be viable. So this shows you that whole thing start to finish. Paracentric inversions have been important in our ancestry. So here's our chromosome 4 and the chromosome 4 of our closest living relative on the planet, the chimpanzee, and they differ by a single pericentric inversion. We don't know which one is ancestral or which one is the evolved condition because, of course, chromosomes don't fossilize. On to translocations. So exchange of chromosomal segments between non-homologous chromosomes, usually reciprocal in nature. You see new linkage groups being formed by them. Position effects can occur from translocation, semi-sterility, and then also the disruption of linkage groups or co-adapted gene complexes. And individuals that are heterozygous for translocations form these cross-shaped double tetrads. That means in meiosis, 4 chromosomes have to synapse, not just two, not just one homologous pair, but 2 homologous pairs, so a total of 4 chromosomes synapsing together, as we can see here. So at the top, we see the chromosomes with the centromeres numbered 1 through 4. The chromosome with centromere 1 and the chromosome with centromere 4 are normal, but the chromosomes with centromeres 2 and 3 have had translocations. You can see the ends are different colored. And then to pair up in meiosis properly, they have to do what they're doing at the bottom there. So the problem comes not so much with crossing over. We're not going to look at crossing over. We're going to look at segregation. How do those 4 chromosomes separate to form gametes, and what's viable and what's inviable? There are 3 different ways they can split up. The one way that they can make viable gametes is called alternate segregation, where the centromeres that are diagonal from each other end up in the same gametes. And there we get balanced or viable gametes. Half of them have the translocation, but they've got all their genes. Adjacent 1 segregation is where the adjacent chromosomes that are not homologous segregate together, and here you get at least 50% sterility with unbalanced gametes that have duplications and deletions. And then adjacent 2, these are when the homologous centromeres segregate together. This is rare because, of course, meiosis isn't supposed to do that. It's supposed to separate the homologs. And so this is unusual, but it also produces inviable gametes. So let's look at these. So here's 2 pairs of chromosomes. N1 and N2 are normal, and their homologs T1 and T2 have the translocations, and that's why they're bicolored. And so for meiosis to occur, you have these 4 chromosomes synapsing like we see at the bottom there. And then over on the left, alternate segregation produces viable gametes. In the center, adjacent 1 segregation produces inviable gametes. And adjacent 2 segregation, which is quite rare, produces also inviable gametes. So here are those things from start to finish. So in a test, I might show you the cross-shaped double tetrad and ask you to split that up in a certain way, and you'd have to show me the gametes, the stuff at the bottom there, and tell me whether they're viable or inviable. So here's a human female. Notice she has only 121 chromosome, it looks like, but in reality she doesn't. If you look at her chromosome 15, it's directly above the 21. You see the one on the right is much bigger than the one on the left. That's because her other chromosome 21 has been translocated or stuck onto chromosome 15. So this person's probably phenotypically normal, but you can see the karyotype is not normal. And sometimes chromosomes will fuse or split at the centromeres. These are called Robertsonian translocations. And we can end up with closely related species that differ in chromosome number but have similar DNA content. And so we count the number of chromosome arms in the karyotype and compare that, and that's a good way to compare these things, what's called the fundamental number. So this shows you how it can work. We have some breakpoints on these 2 chromosomes near the centromeres. Those 2 fuse to form a large metacentric chromosome, and then there's a small fragment left that may or may not be gotten rid of. It can also go the other way, where a large metacentric chromosome, for instance, can split into 2 smaller ones. So muntjacs are small deer from the old world, referred to as barking deer. Reeves muntjac lives in Africa and has only 6 big chromosomes, where the Indian muntjac, of course, lives in India and has 46 much smaller chromosomes. But interestingly, these guys can hybridize in captivity, and their hybrids can even be fertile. It's crazy when you think about the different numbers of chromosomes they're getting from the parents. But these hybrids produce groups of chromosomes that synapse together in meiosis, called multivalents as opposed to ambivalent. So a single Reeves Muntjac chromosome can synapse with up to 5 Indian Muntjac chromosomes. So here are the two species with their karyotypes. Showing you the Indian Muntjac male there and Reeves Muntjac above it with those big chromosomes. And so in meiosis in a hybrid, then one Reeves Muntjac chromosome can synapse with up to 5 Indian Muntjac chromosomes. Now in our evolutionary history, we're of course 2n equals 46. The great apes are 2n equals 48. And the difference there in numbers is the fact that our chromosome number 2 in them is 2 separate chromosomes. So again, we don't know which trait is ancestral or descendant, but clearly there was chromosomes fusing or breaking in our evolutionary history. Now, unequal crossing over, this is the misalignment of the homologous chromosomes during synapsis. It results in duplications and deletions of small adjacent regions of chromosomes. In Drosophila fruit flies, there's something called the bar eye. So a duplication of a small region on the X chromosome causes their eyes to become smaller and bar-shaped. Normally the compound eye in these flies has about 800 little facets, or ommatidia is the official term for that. And flies with the bar eye have less than half, about 350. There's a double bar allele, which is 3 copies of that gene, and that makes even smaller eyes. So let's look at a picture of these things. So here are a couple of wild-type chromosomes in the upper left that have misaligned, and they undergo crossing over, and the red region's deleted in one and duplicated in the other homologue, and that forms the bar allele. And then if these bar chromosomes misalign again, they can form the double bar allele, which is 3 copies of the gene. And here's what it does. So wild-type B++ fly has about 800 facets to its compound eye. One that's heterozygous for the bar allele has 360. One that's homozygous for the bar allele has only 70. And one that's heterozygous for the double bar allele has only 45. So really small eyes. And here's a photo of a bar eye fly on the left, and a wild type fly on the right, showing you how much smaller that eye is. So this shows you a duplication heterozygote, again, where there's a meiotic bulge. So let's now look at changes in chromosome numbers. We're not dealing with the structure of individual chromosomes anymore, but the numbers of chromosomes. Aneuploidy is extra or missing individual chromosomes. And it comes from non-disjunction, which is spindle fiber failure during meiosis, or I guess it can happen in mitosis too. Polyploidy then is extra sets of chromosomes. And it can come about really in 2 ways: allopolyploidy, where we have 2 different species hybridizing, and then chromosomal duplication occurring in the hybrid organism, or autopolyploidy, which is chromosomal duplication within a single species. So this shows non-disjunction. Over on the right here, it can occur during mitosis. If it occurs during meiosis 1, you're much less likely to get to any normal gametes. If you're normal zygotes, if it occurs during meiosis 2, it's possible to get some normal gametes. But you can see here, from meiosis 1, you get trisomic and monosomic zygotes, and from the other, you get trisomic and monosomic, but you can also get some normal diploid zygotes as well. So here's a spindle apparatus attached to the chromosomes. Remember, the chromosomes are the pale things in the middle, and the things attached to them are the spindle fibers. And if they break, that can cause non-disjunction. Spindle fibers. An aneuploid hybrid is the mule. And we know mules are sterile. The reason they're sterile is because they have 63 chromosomes, because horses have 64 and donkeys have 62. And so meiosis breaks down with an uneven number. And interestingly, technically a mule is a female horse cross with a male donkey. The reciprocal cross, a male horse cross with a female donkey, is referred to as a hinny, but I think most people just refer to them all as mules. And here's a little pedigree showing that there was a mule named Kraus who actually was able to produce a fertile egg, and that was fertilized by her father, a donkey, to yield Blue Moon, which was a male mule. So once in a while, a mule can have a baby. but it's rare. On to polyploidy, which is extra sets of chromosomes. So a lot of organisms can't handle that. Having 3 sets of chromosomes is called triploid, and triploid humans die at birth from complete organ system failure. Our systems just can't handle having an extra set of chromosomes. Most times, though, those babies or those fetuses are aborted because they're just too abnormal to develop all the way to birth. So it's rare that a triploid baby is even born. And of course, genetic sex determination can be messed up with extra sets of chromosomes. Polyploids produce often unbalanced gametes. There's a 50% chance of aneuploidy for every chromosome during meiosis, and only 1/2 to the n probability of a normal gamete, where n is a haploid number. So for us humans, that'd be 1/2 to the 23rd, an extremely small number. A tetraploid is better than triploid because even numbers, the chromosomes can pair up together. And this shows some meiotic problems in triploids. Either there's no pairing among the 3 chromosomes, they may all pair up together, or 2 may pair up together, leaving a 3rd one out. And either way, that's often going to cause messed up gametes, either diploid gametes, sometimes triploid gametes, sometimes monosomic gametes, etc. So there can be some messed up things, although sometimes it can work out okay as well. So on to autopolyploidy. So this can come about by fusion of unreduced like diploid gametes or by somatic doubling. In somatic doubling, the doubling of the chromosomes in a cell, in a somatic cell, can happen spontaneously, or it can happen by interruption of the cell cycle through temperature shock to the cells or maybe physical shock to the cells while they're trying to divide. And a lot of our fruits and vegetables that we eat are polyploids, like bananas are triploid and sterile. Sweet potatoes are hexaploid, have 6 sets of chromosomes. Around here, we have these cute little gray tree frogs, and they are diploid, 2n = 24, but there's also a tetraploid species, 4n = 48. The 2 species are identical looking, but different calls. I'll show you pictures in a minute. So here's autopolyploidy. You see it in mitosis above, and then in meiosis down below. So the sister chromatids don't separate like they're supposed to, and so we end up with duplicated chromosomes in the gametes, and then when that gets fertilized, you get triploids, as we see in the lower right. So here's a little gray tree frog. They can turn green as well and have these light spots at the back of the thigh and a light spot under the eye. That's how you tell that species, but there are diploid and tetraploid species, and sometimes they even breed in the same ponds, but because they have different calls, they've never been known to hybridize. The hybrids would probably be triploid and sterile anyway. Now, with allopolyploidy, you get a hybrid from unreduced gametes, or you get somatic doubling in the hybrid. So, for instance, plants hybridize a lot. They cross cabbages and radishes, which are both 2n = 18, and they got what's called an allodiploid. So it was 2n = 18, but it had 9 radish chromosomes and 9 cabbage chromosomes. It was sterile because those chromosomes aren't homologous, and meiosis couldn't happen. But it underwent somatic doubling to become an allotetraploid at 4n = 36. So now it's got a complete diploid set of radish chromosomes and a complete diploid set of cabbage chromosomes. Triticum is wheat, and the haploid number in that genus is 7. And so there are diploid, tetraploid, and hexaploid species of wheat. In fact, the wheat that we depend on for our bread is hexaploid. So let's look at that. So we can cross einkorn wheat with a wild grass and get a hybrid that's 2n = 14, but it's got 7 chromosomes from each species. It can undergo somatic doubling via mitotic non-disjunction to form a 4n = 28, so an allotetraploid that would be then fertile. And when that's crossed with another species of wild grass, Triticum tauci, it's also 2n = 14. We get a hybrid that is triploid, 3n = 21, with a set of 7 chromosomes from each of the species. But when that undergoes mitotic disjunction, it produces bread wheat, Triticum aestivum, which is 6n = 42. So it is a hexaploid. This shows that whole thing start to finish. Some of my favorite creatures are reptiles. And in the Southwest, there are these whiptail lizards that hybridize a lot. And some of these hybrids are polyploid, and they're also unisexual or all female species. So 2 of these species, Tigris and Septem vittatus, hybridized to produce one called the checkered whiptail, which is all female. And some checkered whiptails will mate with males of a 3rd species, Sex lineatus, to produce triploid tessellatus, which again are all female. No male tessellatus have ever been found. And they're more common in disturbed habitats in the Southwest, like road cuts or something, where we've sort of messed up the ecological boundaries between the different species of lizards. So the checkered whiptail is the one in the middle, the tiger whiptail on the right, the other species on the left there. Now we think that they have to undergo this behavior called pseudocopulation. So these are 2 female lizards. The one that's biting, whose face we can't see, that's the one that's fulfilling the male role in this behavior, and it's already ovulated for the season. The one that's looking at us is fulfilling the female role in this behavior, and it has not ovulated. This behavior will cause it to ovulate. So polyploidy is much more common in plants than in animals, and let's examine why. First of all, a lot of our fruits, triploid seedless watermelon, of course sterile, so no seeds. Jumbo apples are tetraploid. Big strawberries, I think, are octoploid. But plants don't have chromosome sex determination, so that's not messed up. They can reproduce vegetatively or asexually until somatic doubling can occur, and they hybridize more frequently because, of course, they don't choose their mates since they cannot move. So anyway, this shows you some of our important crop plants, many of which are polyploids. So potatoes, bananas, peanuts, and sweet potatoes are all autopolyploids, ranging from triploid to hexaploid. And then tobacco, cotton, wheat, oats, sugarcane, and strawberry are all allopolyploids, with strawberries and sugarcane octoploids, tobacco, cotton, tetraploids, and wheat and oats hexaploids. So from chapter 8, do these 3 problems, 22, 37, and 43. And with that, we are finished.